So let's go over the math questions on this AP. So we're starting with 121, and the numbers are weird in the older test, and I think the new, you're going to be switching gears from the first 60 questions into a different part of your Scantron, and we're going to go over these 5 grid-in math questions, okay? So we have researchers observing a large population of birds on a remote island. Birds in a population are found to either have red crest feathers or white crest feathers. So already we're talking about some phenotypes here. And it tells me that the allele for the red crest feather is dominant, okay? And the allele for the white crest feathers is dominant over the white crest feathers. So when I look and see, I can see that the red crest is in larger quantity. Okay, that's my dominant, or at least my dominant allele is there. And then I can see this must be my recessive, and there's the white crest feathers there. So, okay, so pretty simple setup. I have my red and white, my 2 alleles, and these are the number of individuals. And it's important when you do some Hardy-Weinberg to understand the difference between the number of the actual alleles and the number of the individuals, because it can get confusing. Now, they assume the bird is in Hardy-Weinberg equilibrium, so we can use the formula given to us. What proportion of the future populations is expected to be heterozygous for the allele controlling the crest feather color? Well, okay, so the red crest feathers are dominant. And if we want to do that as R, okay, we know that. Now, if we have this many individuals that are red crest feathers, these individuals could be RR, homozygous dominant, or they could be heterozygous. Okay, I don't know. So all those individuals there are the 11,088. Okay, now this, which is really important, that's why the recessive allele is the one you really want to jump at, because unlike what we've talked about earlier about the dominant trait, we can have 2 different, okay, genotypes for the same phenotype. Remember, the phenotype is called the red crest feathers, but the genotype is what the actual alleles are. So if you are a recessive allele like this is, and we know that because they told me that in the setup, that to be a white feather I have to be, I can only be what? Homozygous, homozygous recessive. So that is important. So this tells me something. This value right here represents all the individuals who are this genotype and this phenotype. That's the key. So I can't jump with the dominance. This one I'm going to use. Now, if you go to your reference table, and let's do that, okay, you should realize that there is some things we can look at. And in the reference table, they tell us very nicely about the Hardy-Weinberg equations. Okay, and there we are, in all of its glory. Okay, we can see that the frequency of the dominant allele in a population, and Q is the frequency of the recessive allele. Now what they're not telling you is that this represents the what? Individuals. P squared would be what? Dominant and dominant. So if I have P squared, that must be all of the homozygous recessive. In the case of my red crested feathers, this would be big R and big R. This would be all the individuals that are homozygous recessive, little r, little r. Okay, so that's where the Q squared comes from, and the P squared, 2PQ, is all the individuals that are heterozygous. So what we're after in this problem, we're after the individual number of alleles that are dominant and recessive. So let's go back to the problem. Okay, so here I go. Now, in order to get the P and Q, because what they're asking me is give me the future or proportion of the future population expected to be heterozygous. Okay, so we're looking for big R, little r, and we should know that's 2PQ. That's what we're after. So if we look at the equation that's been given to us in our reference table, so from the reference table we have P squared plus 2PQ plus Q squared. And all this tells me is that the frequency, or the percentages, if you want to think about it, all equal, of course, 1. So this is the frequency, or percentages, or fractions, or decimals that have to equal 1 from all the individuals. Like I just said, this is going to be R and R. This will be R and little r. And this, I'll put Q there. I'm thinking R, but knowing that's Q. Little r, and this is R, R. So we know that the Q is the recessive, and we know that the P is the dominant. So I'm looking for these individuals. I'm looking for that fraction of 1 that those individuals will be considered, knowing these values here. Okay, so let's get to work here. All right, so what do I know? Well, the first launching pad here is the recessive. The recessive tells me, as I just said, something about the Q squared, the individuals that are here. So that's my launching off pad. So how do I get this percentage? Well, from these individuals, I know that it's a fraction. So what I do is I do part over total. So I'm going to take the white crested feathers, which are 1,759, and I'm going to divide that by the total, which is going to be 11,088 plus 1,759. And that's going to give me the frequency, or the percentage, if you want to think about it, the fraction of the recessive alleles. So putting that in my calculator, 1,759 divide by 11088 plus, big finger error, plus 1759, close my parentheses, and I get a frequency to be, where am I, 0.137. And what does that equal? That equals not P, that equals, or Q, it's Q squared, right? This is the frequency of the recessive over the total. So that's Q squared. All right, those are the people. Why it's the people? Because these represent the R. R times R is recessive times recessive is Q squared. That's what we have here. So we want to find Q. Why do I want to find Q? Because that'll get me P. So what I'm going to do is I'm going to square root both sides. And what that gives me is Q is equal to the square root of that. So let me do that in my calculator. Square root of that number, and I get Q to equal 0.37. Okay, great. How do I get P? Well, if it's all the alleles in the universe are either little r or big R, if I have 0.37, I just do 1 minus 0.37. All right, and I get what? I get 0.63. And that equals my P. Now I can do my 2PQ. My 2PQ, and let's make some room here. My 2PQ, very simple. Is equal to 2 times my P. My P is 0.63. Ooh, color coding. Times my what? My Q, which is 0.37. And it's no accident that they equal 1, right? P plus Q equals 1. That's why this all works. There's only those combinations. So let's put this in our calculator, and we get 2 times 0.63 times 0.37, and I get 0.4, rounding off 0.47. They say round to 2 decimal places, so I get 0.4662, but because I'm rounding to 2 decimal places, I want to round to this number here, but I look at the number next to it's above 5, so this becomes 0.47, okay? And that is my answer. And that's the frequency of people that are heterozygous, assuming we're in Hardy-Weinberg equilibrium, and these are the current number of individuals showing these phenotypes, okay, in our population. So let's go on to the next one. So number 121, a typical human lymphocyte, we should recognize that. It's a white blood cell, has a radius of 10 micrometers. It's not important that you know that symbol, but let's draw a round cell, and let's say its radius, okay, is 10. While a tick of bacterium, okay, has a radius equal to 1. So that'll be much smaller radius, and that'll be 1. Okay, so what we're going to do now is figure out, assuming that both cells are perfectly spherical, how many more times is the surface area of a typical human lymphocyte compared to? This is going to be bigger, but how much? You would think maybe 10, but we need a surface area. Okay, so there must be a surface area formula to look up. Okay, we had that lab that we looked up, so let's go there. Okay, so we're back at our reference tables, and we're not going to go there. Okay, let's go to our reference tables, and if we go to the back side, toward the bottom, we're going to see our surface area formulas. And where's our surface area of a sphere? Right here, 4 pi r squared. Okay, so we're going to use that. So the surface area of a sphere is equal to 4 pi r squared. Now we can calculate this whole thing, but let's see what we have here. So the formula here is going to be 4 pi r squared. The radius is 10 squared. And that's going to equal 10 squared is 100 times 4. That's 400 pi. And I could put that number in, but I know it's 400 of these units if you want to think of it that way. And the next one is 4 pi, and of course in this case the radius is 1, so 1 squared. So that'd be 1 squared is 1 times pi times 4 is going to be 4 pi. So my question is, now that we have solved for surface area, you may say, Mr. Grotsky, why didn't you just times it by pi? I don't have to. Okay, if you think about it, yes, if I want to know the exact number, I would have to take it by 3.14. But since they both have that factor in, can I just compare them? Isn't 400 bigger than 4 by 100 times? And if I'm not sure, take my 400 pi and divide it by the smaller 4 pi. And for those that like the math, you'll see the pi's cancel. And 400 divided by 4 is equal to 100. So you would say that this cell has 100 times more surface area than the bacterium. Okay, and again, the whole key of this was we had to look in the reference table for our surface area for a sphere. Okay, we did that one lab there. Okay, moving on to number 123. This is a formula that looks strange to us. We have Tm equals 4 times G plus C plus 2 times A plus T. So they're giving us this formula, and they're basically saying this foreign formula that you haven't seen, can you just follow it with some kind of biological connection? And the connection here is we have a strand of DNA with the appropriate nucleotides, and they want to predict the average temperature, and obviously there's some way to do that using the strands of DNA in this formula. So they want you to use in the formula what's the predicted melting temperature, okay, for this single strand of DNA using this formula. So basically Tm, okay, which is the melting temperature of the strand, is equal to 4 times the sum of the guanines and cytosines. So let's count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Make sure I got all of those. Didn't miss any G's or C's. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. I got 11 of those. That's total. Okay, so 4 times 11 plus 2 times the number of adenine and thymines. So let's do a different color because I can. Here's 1, 2, 3, 4, 5, 6, 7, 8. So the sum of all the adenine and thymine are here. And so I have, just following the formula, again, I know we haven't seen this formula, but I think we can handle this. 4 times 11 is 44. Plus 2 times 8 is 16. Okay, and you put that together, and what you get? And you'll get 60. And they didn't ask for any rounding or anything, so 60 is a number. Okay, it's pretty straightforward. I know it sounds weird. We haven't seen this before, but That's what they make you do sometimes. Let's go on to the next one. Okay, this is a graph question. And so we basically have the, I guess, amount of light that's penetrating the water depth. And obviously, as we get deeper, there's less light. And what they're asking us in this question is, what is the deepest water level? So what's really the water depth? Okay, in which the light penetration is expected to be at least 200. And like, don't get crazy about what these units mean. Okay, so let's go find 200. Where's 200? Right here. And they're just basically saying to you, what do you think that value is right there? Okay, well, I've got to be able to interpret this graph. If this is 5, this is 4, right? This must be 3. This is 2. This is 1. So it looks like to me I'm like 1.6. All right, and give me an answer to the nearest tenth. Okay, well, this is already in meters, and this is single digits. This is the tenth, so 1.6 is my guess. Now, they're going to give you a range. I mean, if you thought it was 1.4 or 1.7, they're giving you a healthy range to guess there. I believe it's 1.4 to 1.8, but I'm not mistaken. But so you can see that that's certainly something in there. And again, looking at it again, it's maybe more like 1.4 than 1.6 to me, because it's my straight line is kind of crooked. So that's why you're given some leeway. So I believe it's 1.4 to some range. So any of these numbers that you interpret, and I guess looking at this again, maybe 1.5 is better, who knows? But even that, all these numbers would work. As I put this in your form, I gave you the range, I believe, from, I forgot what the exact range was, but these would all be acceptable values for just reading what value is your x given a certain value of y. That's all this question is, guys. And they give you some plus and minus room to wiggle there. All right, so moving on. And let's go on to the last question. And again, these aren't very difficult, maybe weird to you, but here's what they have here. They gave you a little setup here, but I'm getting right to the math. Based on the information in the graph, how many more times are ants found on plants exposed to solvent plus chemical compared? All right, so we're going to basically compare. This is the solvent plus chemical line. So how many more times are ants that are on plants exposed to the solvent plus chemical? So solvent plus chemical. All right, that's what we're doing here. That's where this graph is. Compared to plants exposed to the solvent only. So we're comparing these two. Based on the information, how many more times? It's a ratio. So watch what I do here. Very simple. Let's go find the smaller value. Okay, the smaller value here. And again, don't have a straight edge. I'm just kind of guessing. But it looks to me like I'm somewhere in between these two numbers. So I'm going to guess that my value, okay, is somewhere around, okay, between 35 and 40. So let's say 37. And what's the value of the solvent plus chemical? Well, not quite 90, so I'll do 89. So how do I find the ratio? Well, big over small. So I'll do 89 divided by 37 in my calculator. And here we go, 89 divided by 37. And I get 2.4. And that's my ratio. Very simple. They would give you they'd give you a range of those based on what you guess for those values. But that's going to be your ratio, or your how many more times. So 89 is 2.4 times bigger than 37. And that's it. And again, you would have some room to wiggle. There'd be plus or minus. And I'm not sure exactly what the plus or minus is. I put that in your form to figure it out. But you should have some room to wiggle because it's hard to get exactly what these values are. So there'd be a range of answers that make sense, maybe from 2.1 to 2.8 or something. But I'm coming up with 2.4. And just always check to make sure that you are giving it in the proper degree of precision, as we would call it. And we are going to 1/10th place. So 2.4 would be my answer. Hope that helped.